Petra. I'm a fourth-year PhD student at Emmanuel College, and I do my research at MRC Mitochondrial Biology Unit. So I'm originally from Slovakia, and that's where I did my high school studies. I first did the half-a-year internship in Cambridge. During this time, I secured a PhD at MRC MBU and became a member of Emmanuel. So depending how much biology you can remember from school, you might know some of this already, but I'll help jog your memory. All eukaryotes, which include all the large organisms you might think of, have cells that contain a nucleus and other microscopic structures called organelles. Mitochondria are one of these organelles inside almost all eukaryotic cells. In humans, they are maternally inherited. Their main role is the production of cellular energy, which is stored in a compound called adenosine triphosphate, or ATP. The most widely accepted theory of the origins of mitochondria is that bacteria became endosymbionts living within the cells of other organisms, which over time evolved into the mitochondria we have today. While most people think of the DNA we have in our cells existing in the nucleus, which is where most of it is, mitochondria also have their own DNA. Something needs to happen with this information, and for that it's the process called gene expression, which is transcribing RNA from DNA and then translating proteins from this RNA, and then the proteins perform all the functions within the cell. This is what mitochondrial genetics studies, and this is my field of research. In doing research, it's important to find your motivation and know kind of what drives you, especially in the situations when things don't work out. And in my case, it's curiosity. I'm really eager to learn about how things work and kind of broaden our knowledge of cell biology. There are around 20,000 genes encoded in human DNA in the nucleus. Human mitochondria DNA contains only 13 protein-coding genes. Although this number might seem small, each one of these proteins is crucial for proper function of mitochondria and therefore securing enough energy for the cell and the organism. The method we use for investigating the roles of specific genes is to turn them off, which is called producing a knockout. Then study what happens with the cell physiology or look at the molecular changes afterwards. A knockout of a particular gene might produce a change in the growth rate of the cell or make it dependent on certain nutrients, for example. This helps us to pinpoint the function of different genes. On a molecular level, we can assess whether cells are still able to synthesize mitochondrial proteins. We can look at overall changes in levels of all other proteins in the knockout cell line. By observing these changes in the cell functions, we can pinpoint the process in which the gene of interest is involved, as well as better understand the process itself. Proper function of mitochondria is very important for the cell to be able to survive. Mitochondrial diseases is a very broad range of diseases. They tend to affect mostly tissues that are very energy demanding, such as neurons or muscles. But they've been also implicated as a factor in human diseases such as Alzheimer's, Parkinson's, or type 2 diabetes. At the moment, there are no cures for mitochondrial dysfunction. Knowing what is going on, I think this is kind of the first step towards the ultimate goal to find the cures. We can verify whether the variant of a gene that's identified in a patient is consistent with what we see in a knockout cell line, for example. I think it's really interesting when you're doing these experiments that maybe don't seem to be too much related or seem to be a set of individual results, but then you kind of look into that from a different angle or do this one other experiment that kind of suddenly everything makes sense and it comes together, and it's about being able to understand what your results mean is the bit that's really rewarding.